Sickle cell disease. Sickle cell disease is a hemoglobinopathy. So it's a disorder of the hemoglobin. But in this section, our focus is on 5 levels of prevention, how we can prevent this condition in the community or in the public or in the family following the 5 levels of prevention. So, sickle cell disease is a genetic disorder characterized by abnormal hemoglobin, which cause the distortion of red blood cells into a crescent shaped or sickle shaped shape. This disease is caused by a mutation or a distortion that affects the gene that is responsible for the production of globin chains of the hemoglobin molecule. Normally, a hemoglobin is composed of 2 alpha and 2 non-alpha globin chains. It is worthy of note that alpha globin chains are produced throughout life. So therefore, severe distortions while in the womb can cause intrauterine death. However, production of non-alpha chains varies with age. Varies with what? Age. So, what is hemoglobin? Hemoglobin is a very important protein that is found inside the red blood cell. This is the red blood cell. Its main job is to carry oxygen from the lungs to all parts of the body and return carbon dioxide from the body back to the lungs. Hemoglobin, like we said earlier, is made up of different proteins called globin chains. And these globin chains is of 2, alpha and beta chains. Why do we say that sickle cell disease is a hemoglobinopathy? And what are hemoglobinopathies? Hemoglobinopathies are disorders that affect the hemoglobin itself due to distortions or changes in the genes we mentioned above. These changes occur in these globin chains. When these mutations or changes occur, it can now lead to abnormal production of hemoglobin, which is the major cause of this sickle cell disease. Types of hemoglobin. Types of hemoglobin found in this red blood cell. Each red blood cell is made up of hemoglobin. Inside of it is hemoglobin is found here. Hemoglobin is found here. Hemoglobin is found here. Also in this sickled one, this is a sickled one, the sickled red blood cells. The hemoglobin is found inside. We have 2 types of hemoglobin in human being. We have the fetal hemoglobin. This is the type of hemoglobin that you find inside the fetus, when the fetus is inside the womb. And that type of hemoglobin has only 2 alpha chains and 2 gamma chains. Whereas adult hemoglobin, that is the hemoglobin that is in the red blood cell of an adult after being born and throughout adulthood. And this hemoglobin has 2 alpha and 2 beta chains. But remember, in fetal hemoglobin, it has 2 alpha and 2 gamma chains. But in adulthood, it has two beta chains, two alpha chains, sorry, and two beta chains. Why do we need to understand this? The reason is because after 6 months of age, that is, when you give birth to a baby, remember the baby's hemoglobin has two alpha and two gamma chains, but adult has two alpha and two beta, so they are different. So what it means is that it's only after 6 months that hemoglobinopathies like the sickle cell can be seen. Because after 6 months of age, that is when the fetal hemoglobin would have totally be replaced with adult hemoglobin. So therefore, if there's a problem with any of the beta gamma chain, the beta chains, sorry, or the alpha chains, no, mostly the beta chains, because the alpha is in that of the fetus and also in that of the adults. So if there's any problem in the beta chain, it is then after 6 months of life that that condition will show up. Now let's also look at how does this sickle cell happen. This sickle cell disease happen through the changes in the hemoglobin. First, hemoglobinopathies, which you all know that issues with hemoglobin, we can group it into two: qualitative abnormalities and quantitative abnormalities. When we say qualitative abnormalities, we are trying to refer to those changes that happen to the globin chain. Remember, it's only on the globin chains. This means that the hemoglobin molecules might be present but are not working properly. For instance, in sickle cell disease, there is a specific distortion where one amino acid, that is, you know that amino acid is the building block of protein, is being replaced with another in the beta chain of the hemoglobin. So that replacement causes the problem in the gene. That is why sickle cell disease is a genetic related issue. It happens at the genetic level. Whereas other hemoglobinopathies can also be grouped as quantitative. This happens when there is either overproduction or underproduction of one type of globin. Maybe in that sequence, that particular protein is supposed to, or that particular amino acid is supposed to be 20, but now it's not 40. That unnecessary increase, that distortion can lead to hemoglobinopathy. So this is an overview of the genetic part of sickle cell disease, because it's good we understand that sickle cell disease has a genetic root. So when we are preventing, we focus more on primordial level so that we can actually prevent the disease. Still going forward, in sickle cell anemia or in sickle cell disease, the problem there is that there is a single change in the amino acid sequence of the beta chain of the hemoglobin. What happened actually is that the sixth amino acid is changed from glutamic acid to valine. So that single substitution, qualitative substitution, is what leads to sickle cell disease or sickle cell disorder in a person. Now, if you look at this picture here now, These are normal red blood cells. These are normal red blood cells. See how round it is? See how round it is? But see these ones, these are those ones that are having that abnormality due to quantitative or qualitative related issues that will now make the structure not to be resilient enough when it passes, when you have deformed, it can no longer return to that shape, and thereby reducing the oxygen carrying capacity of these red blood cells. These red blood cells are meant to carry oxygen. Look at them. This is a white blood cell, and this is an artery where they are where they are flowing, carrying oxygen along to other cells. But these ones now are already sickled. And as they are sickled here, they can stick together and block blood flow, which is the major cause of sickle cell crisis and symptoms. And also, if there's infection, let's say there's an infection now here, and blood rushes to that place, and these sickles are there, they will block the normal blood flow to this place, and the inflammatory responses and all of that will amplify, and more will not cluster, leading to crisis. That is why in sickle cell patients, they present with serious pain, they present with jaundice. Now, how did jaundice come about? Because these are now abnormal, the body will see it as abnormal cells and trigger a reaction that will destroy them. And once they are destroyed or being hemolyzed, the person will become anemic because the total number of red blood cells has decreased. Let's say this is the total number of red blood cells in this body, and this is destroyed. It has already been reduced, and the person becomes anemic and presents with jaundice and other signs and symptoms of anemia. So that is what really happens in a patient with sickle cell disease. So the first level of prevention is the primordial prevention. Primordial prevention focuses on preventing the emergence of risk factors in the population who are healthy. So the main target of primordial prevention is to make sure that nobody is exposed to a risk. And you know that a risk is something that makes you more vulnerable to have a disease. It's something that makes you more common or more opportune to have a particular disease. So in the context of this sickle cell disease, having the gene alone have predisposed you to having sickle cell anemia, especially when you have it from both parents. So the first thing we do is to make sure that people are not even exposed or people don't have the risk. And that is where we as healthcare workers come in. And what do we do to ensure that? Education of communities. We do education and create awareness. And in that education and awareness, what is our focus? We are going to let the community and people to know how sickle cell is being transmitted from parents who are carriers. From parents who are carriers. We must emphasize that it is autosomal in nature, autosomal recessive in nature, which means that before you can have, say you are fully a sickle cell patient, it means that your parents, let's say your dad has S for the normal and the smaller S for the abnormal. Then your mother has bigger S for the normal and small s for the abnormal, which means your dad and your mom are carriers of sickle cell. So therefore, you can only have it if you inherited this smaller s together. You inherited the abnormal one from your father and abnormal one from your mother. You will not have the sickle cell issue. But if you inherited the good one from your father and inherited the good one from your father, from the bad one from your mother, you can only become a carrier. You cannot be a full-time sickle cell disease. So what we mean by autosomal is that you must have gotten the abnormal gene from your father and also the abnormal gene from your mother. But if you have the one good one, you cannot have the disease, but it can be a healthy carrier of the what? The disease. So that is what we mean autosomal recessive. And also we have to educate the community that if you already have you are at the career status, you must not marry with another person who is also a carrier, so that your offspring will be saved from the crisis or the chances or the after effect of being having sickle cell gene, and the implication as a mother, and as a parent caring for a child who is sickle cell disease patient. Another thing we do at primordial level is genetic counseling. There's importance and there is need in educating individuals, especially in regions where there is high prevalence. There is high prevalence of this disease. So you tell them, especially we in the African region, especially in Nigeria here, we are exposed to this condition. So it is important they go for every couple before they get married, they go for genetic what? Counseling, so that they can reduce the risk of having children with sickle cell disease. Now, the next level of prevention is the primary prevention. We apply primary prevention when the individual is already exposed to the risk. The person is already exposed to the risk, but we can help the person from not coming down with the disease. So primary prevention aims to prevent the onset of the disease in an individual who is already at risk. And what activities we do? Prenatal screening and diagnosis. So under this level, what we do is we offer prospective parents, those parents who already have the gene, screening programs to identify those at risk of having children with sickle cell disease. So we do prenatal screening and what? Diagnosis, in order to identify the next level of prevention is secondary prevention. In secondary prevention, it involves early detection and prompt treatment to manage existing disease effectively and prevent complication. Here, the person is already having the disease. So what do? We'll continue to do routine screening, regular health checkups, and other screening to detect early signs of the complications in individuals who are already diagnosed with. For example, monitoring the hemoglobin levels and also the blood viscosity and organ functions. So these are what is done at this level. The next is, okay, continue under the same secondary prevention. You also talk about vaccination of these children. It's important to vaccinate children with sickle cell disease so that they will be protected against common infections such as pneumococcus, meningococcus, and Haemophilus influenza type B. This will help reduce disease-related complications because most of these conditions affect them because it usually trigger immune responses. And you know, immunity and all that is involved will lead to excess blood flow and release of some mediators, which will help in the coagulation or this sickling of most of the red blood cells into in the vessels. And when this happens, most of this can lead to crisis that is being seen in a patient with sickle cell disease. Another thing that is done, the secondary prevention, is antibiotics prophylaxis. It's always important to offer penicillin prophylaxis to children with sickle cell disease during the first few years of life, so that to prevent them having infections, particularly pneumococcal infections, which are more common in these individuals. So you know that the most important thing is to prevent them from coming down with these infections, because the immunological response, everything that happens, will definitely trigger the crisis. And the processes and the bioprocesses, biological reactions that take place, disease fighting processes, will definitely lead to destruction of more red blood cells, as these red blood cells will rush to the site and they will lose the ability to return back to normal shape, and there will be lots of them being destroyed. Then hydroxyurea therapy can also be given. When you administer hydroxyurea therapy, can help increase the fetal hemoglobin levels and reduce the frequency of pain crisis and vaso-occlusion events. What we mean by vaso-occlusion events is when those red blood cells are destroyed, or as they are formed in seckled form, they tend to block the vessels. And when that vaso-occlusion happens, blood flow will be impeded, and as such, the person will come down with a lot of crises, pains, and all of that. Now, the next level of prevention is tertiary prevention. This level focuses on managing and treating the disease effects in order to improve quality of life and minimize complications. So the essence is just to make sure that the person who's already having this condition and the complications is able to live a quality life. Therefore, the essence is to minimize the complication the person is having. And this involves comprehensive management of the patients, which includes pain management, which we do, mental health support, trying to talk to the person can have a better life, live a better life. Then routine medical follow-up for these individuals with this condition. Then blood transfusions. Regular blood transfusions may also minimize severe complications, including acute chest syndrome. We will discuss about acute chest syndrome and stroke more when we are talking in details about this sickle cell disease, how it happens, what happens in the body. But with a brief we've been able to look at, I believe you are understanding what would warrant the giving of blood. Remember, the sickle cell red blood cells, once they are in that sickle form, they can no longer function very well, and the body would likely destroy them and reducing the number of the red blood cells. And when that happens, anemia will set in. And so there is need for blood transfusion. Another thing that's done at tertiary level of prevention is bone marrow transplant. Although this is for eligible individuals, where hematopoietic stem cells are now transplanted into these people in order to save them. It can also be the cure for sickle cell disease, because these stem cells will now be producing healthy red blood cells that do not have that background of distorted beta globin chains. Another thing we do is also education of these patients on crisis management, how they manage their crisis, such as pain crisis. Also educate them on hydration, that is taking enough water. Remember, if they take enough water, the plasma volume will increase. As such, the chances of the cells coming together, being clogged and occluding a vessel in their sickled shape would be reduced. Also, we educate them on rest. They need to take enough rest and avoid extreme temperatures like cold that can trigger immune response or body response or allergic response. Then excessive temperatures like overheat and all that can trigger a lot of um, responses for body adjustments. To tell them to avoid that. Then the next level is quaternary level. Quaternary level prevention is focused on preventing over-medicalization and unnecessary interventions based on patient's need. So the thing that are done there is patient-centered care. It's important to ensure that the treatment plans are personalized, narrowed down to meet with the patient's needs, so that to avoid excessive interventions or treatment that may not really help the patient or it may harm the patient. Then also another thing that is done there is shared decision making. It's always important to engage with the patient so that the patient understands the various treatment options, weigh the benefits and risk, and also ensure they understand their choices and what may likely happen if they do not accept certain options or if they accept certain options of what treatment. That is shared decision. The patient is part and parcel, or the client is part and parcel of the decision being made about his or her condition. So that's all about sickle cell, prevention of sickle cell disease, following the 5 levels of prevention. See you in the next section.